All right, everybody, we're going to go ahead and get started here. Today we are going to be hearing from Brianna Rivera. She is a physical therapy tech in our comprehensive therapy department here at Insight in Flint. She's going to be doing a presentation on a spinal cord injury treatment, a study regarding that done by the Northwestern University. So without further ado, here's Brianna.
Thank you so much, Evan. Well, hello, everybody. I'm Brianna. I've been at Insight in Flint for just under about 3 years now. I never thought I would be giving my own grand rounds, but I'm really excited to be here and to get the word out about this study just because I thought it was so impressive. So I just hope that I can show you why I think this deserves attention today and that you leave maybe just a little bit excited because I know I am. All right. So just before we kind of start talking about the specifics, I just wanted to highlight the research team that made this all possible. And science is never done alone, and this took a lot of people working together to accomplish a really big feat for medicine. So, all right, but what we'll kind of go through today, we'll talk about kind of the main molecular player that causes cellular differentiation and proliferation, which are the peptide amphiphiles, and I'll just be calling them PAs for short because that's hard to say. We'll talk about the challenges that have to be overcome in the healing of a spinal cord injury, the goal of the treatment, how these PAs assemble in solution, what we observe in vitro, what we observe when the spinal cord injuries in live mice are treated, and then the actual behavioral changes that we can see. All right, so to kind of break down the chemistry, I know everybody I work with gets scared when I start talking about chem, but I'll try to make it interesting for you. These tiny little molecules, they are just a few nanometers long. They are actually a part of a class that's called a supramolecular polymer. So it basically just means that they're different polymers stuck together. They were only synthesized back in 1995, and they've been a really big interest in research ever since. Currently, they are being used in medicine. There was a new cancer drug developed for treating neuroendocrine tumors, excuse me. They also have been shown to slow internal hemorrhaging that's not able to be stopped just by applied pressure. But most importantly, they're really studied in regenerative medicine. So the big promise that's locked inside of these nanostructures is that they have this unique ability to assemble into lots of different 1D nanostructures. So things like hydrogels, micelles, and fibers, and they mimic the structure and the function of molecules that are already inside of our body. So the other really big significant piece that comes along with PAs is that we can integrate really specific cellular signals into their body so that they elicit some kind of targeted cellular response better than what our body can provide for us. So the three different polymers that make up this supramolecular polymer, the first piece, which is shown kind of here in segment 1, there we go. It's just really a hydrophobic carbon chain backbone to provide foundation and structure to the molecule. The next piece is this 8 to 30 amino acid sequence. So what you would see in 2 and 3. And specific amino acids are selected because they're the thing that actually influences what structure is going to be formed by these molecules. And then the very last piece, seen in 4 and 5, and kind of this big conglomeration right here is this charged head that's promoting solubility in water. And it's also where we have the biosignals attached in the molecule. All right. So why use peptide amphiphiles for spinal cord injury repair, and what's the significance that they're able to overcome that the body is not? Really, one of the big reasons why the body can't promote regeneration and repair of this type of injury itself is the structure of the actual axons. They're really long and lengthy, so it becomes really difficult to transport all of the necessary molecules for healing to the site of the injury. And the area of injury or repair that has to occur is going to be all the way from the site of the injury back up into the brain. So this neural network has to be completely restructured, and the body's normal rate of nerve regeneration is only 1 inch per month. So it just does not have the capacity to correctly heal this injury. So let's see, sorry. We do know of 2 signals that have been shown in other studies to elicit both cellular differentiation, stem cell differentiation into neurons, and cellular proliferation. So the creation of new neurons and also just the growth of new cells. So our IKVAV is just an amino acid sequence that promotes the differentiation, and then the FGF2 is going to be responsible for proliferation. It's just fibroblast growth factor. So we know that these 2 work well in promoting nerve regeneration, but this has never been tried before for spinal cord injuries. And then there's a few other kind of limitations that we have to account for and attempt to overcome in the structure of the peptide amphiphiles. So one of the big issues, especially with why the body can't produce this regeneration, it kind of comes to like the foundation of chemistry and collision theory. So in order for reactions to occur, we, number one, have to have things colliding with one another, but the collision has to have enough energy and it has to be at the correct orientation for chemical rearrangement to actually be possible. And because the axon structure, it takes a long time to move these things, they don't collide with enough energy or really a high enough concentration. So that's what we're trying to get the peptide amphiphiles to be successful at. The last limitation thermodynamically that occurs that's been observed in PAs in the past is that sometimes there's other pathways of the reaction that are possible, so we don't end up with the desired product that we want. So we want to try to assemble a structure that's going to avoid this thermodynamic trapping. And then very last thing that was considered in the architecture of these molecules is that the 1D structure that they assemble into has to mimic the normal environment of the spinal column, so that number one, the immune system doesn't think this is an intruder and just take over and kill these things. And it also needs to provide the same function as our normal cellular makeup. So with that, we can move into the microscopic and macroscopic hypotheses based off of all of these challenges and all of our knowledge about peptide amphiphiles so far. So the desired structure that we're looking for these things to assemble into is going to be fibers. And they specifically want to mimic collagen fibers that are normally found in this extracellular makeup because they are responsible for cellular adhesion and then cellular survival of these new cells that are being proliferated. We know that we have to include our IKVAV signal, the fibroblast growth factor signal, and then there is another really important piece that has to be, or there's an important property that these have to contain in order actually to promote the correct collisions. So there needs to be a very large chemical potential store within these molecules, specifically within the peptide sequence. And one way that we can really effectively store chemical potential energy is with hydrogen bonding and the formation of beta pleated sheets. So basically, once these molecules are assembled, the protein sequence is kind of going to fold up just like an accordion. And then when this is able to hit water, the water is going to take over and unbind this pleat. So the tighter that you can get these pleats, the more displacement that's going to occur, which is just basic spring theory from physics. All right. So if all of these things go correctly in the microscopic realm, what we should be able to observe macroscopically is structure and functional changes to the damaged tissue and the various biological networks that need to be restored at the site of the injury. We also should observe a significant change in hind limb mobility and function of the hind limbs in live mice subjects. All right, so to look at kind of the first set of results, this is just evaluating 8 different peptide amphiphile structures, how they assemble into 1D nanofibers, and how dynamic are these structures. So what's kept the same in all of these is that carbon backbone. There's also a 5 amino acid sequence that's kept the same. What ends up being different is just one little section that's 4 amino acids long, and they are using valine, valine, alanine, and glutamic acid, so VA and G sequences, because these 3 amino acids provide the highest propensity for beta sheet formation. So in figure A, we've just got the breakdown of different structures. Figure B now is actually analyzing how much mobility these different variations do have. And what's interesting is that we see the forefront PAs being PA2, 5, and kind of 4 is giving us intermediate range of mobility. So the blue areas being super high mobility, the red being very low. And looking to the peptide structure of PA2 and 5, they actually did not contain valine or the V amino acid, and that amino acid actually provides the strongest hydrogen bonding. So it's interesting that out of this first method, we see what the optimal sequence and architecture of these peptide amphiphiles would be. And even though we want something that's going to be able to form a very tight beta pleated sheet, sometimes the intermolecular interactions of the molecules are too strong that the water coming from the outside actually can't pull them apart. So we don't need actually any V amino acid in these sequences. It's only going to be A and G that provide this optimal level of motion in solution. All right. The very last thing to kind of discuss from this first method that I thought was pretty notable is looking at the bar graph in figure C. This is measuring the average displacement of these molecules over a certain amount of time. And it was generated through actual a computer molecular simulation. So we do see in B and A like the actual mobility results, and then in C, the simulation is also further confirming our hypothesis that PA2 and PA5 are going to be the most bioactive compounds and have the highest mobility. All right. So the second method to look at, what the researchers moved to next, was some in vitro treatments of these different peptide amphiphile solutions. So let's see, in vitro is just treatment at the cellular level. So these are just plates of cells that are being treated. We're not into live mice experimentation yet. But we get some very pretty pictures and also very interesting results when looking at the different treatments. So as predicted, our forerunners, which are PA2 and 5, in figures D and F, this cellular staining technique just gives us a bunch of different colors. Red is going to be the stem cells present in the plate. The green is really the significant piece that we're looking at, especially in figure D, because it's just some cellular receptor antibody, but the presence of it means that we have activated the differentiation of the stem cells, and it also is telling us that we've activated proliferation in the cells themselves. So where we see the highest density of green in 2 and 5, we know that these are very good candidates for regenerating neurons and axons. Figure E here, is just a Western blot analysis. So it's reagent testing. All of these are just different cellular antibodies that we know are released when these specific cellular scaffolds occur. And following the results in D, our forerunners 2 and 5 are still the most successful at activating this in the cells. And then in figure F, this one really excited me. The only difference in the color staining that we see is this white network that's appearing, and those are actual neurons. So just in the in vitro experiments, figures 1 and 2 are giving us very strong evidence that what we theorized microscopically is actually possible in this treatment. So now we can kind of start zooming out and looking more macroscopically at the tissue networks and then really at actual mice subjects. Okay, so the next method that was investigated was actual spinal cord injury treatment. So there was both in vitro testing performed on axon regions that had injury occurred to them. And then there was also actual injection into injured mice and some into uninjured mice just as a control. But we actually, from the past results, we start to narrow down the different PA treatments. So because only 2 and 5 really promoted cellular proliferation, we're not going to use any of the other ones. And then between these two, only PA number 2 was able to form not only the fibers, but like a hydrogel solution. And the viscosity of the hydrogel that's formed there really closely mimics the extracellular matrix found in the body. So these things are going to be able to actually mix and combine to react together. The other PAs were not able to do this, so they weren't going to be effective in an actual biological environment. Okay, so 2 different treatments then arise for injection once we've kind of whittled down the different PAs. So one is a mixture of our PA2 with the IKVAV signal. There's also PA1 with the fibroblast growth factor integrated into it. This gives us a mix of like a low and high mobility mixture. There's also just a high mobility mixture with only peptide amphiphile number 2 involved in it. All right. So what happens is, unfortunately, the only sad part about the study is these mice are injured surgically. So they're using a mirroring method and first performing a laminectomy on the mice and then using a tool to kind of compact the spinal cord at a certain place and create a contusion that's going to inhibit hind limb mobility. 24 hours after the contusions are created, that is when the injections occur. So, let's see. The first thing that is investigated can be seen up here for figure D. Sorry, excuse me. Okay, so they first just wanted to see how this PA solution, the treatment, broke down in the actual body. And so in week 2, that little red splotch that you can see in the green spinal column is pretty concentrated. It is pretty notable that they're able to localize around the injury so closely. When we compare that to week 12, so this is 3 months later, there's hardly any PA solution that's even left. And instead of this red splotch where the injury occurs, we just have green spinal tissue. So that alone is very promising because number one, we're seeing some regeneration already, but we also are seeing that this just naturally biodegrades into the body, which is quite useful, so that we don't have a bunch of junk floating around in there. In figure E, this is again just looking at a graphical representation of the biodegradation for all the different treatments, and it's a really nice linear pattern that goes down almost to 0 for the volume after these 12 weeks. All right, in figure F though, this one is one of my favorites from the whole study. Not only are we already kind of seeing repair occur at the site of the injury, figure F is telling us that we also are regenerating the entire neural network from the injury all the way back up into the sensory motor cortex of the brain, and that's where voluntary movement is controlled. So basically the researchers use this tagging method to mark kind of the beginning and the end of an axon pathway, a neural pathway. So that's shown in the red here. So at the site of injury, you can see the little cluster of neurons that are tagged. These go all the way back up here into the sensory motor cortex. So the red that's seen in this stain is showing us evidence that this restoration is performed through a very big section of the spinal column and into the brain. All right, the last kind of thing that comes out of this method was now we really see which treatment is going to be the most effective at restoring structure and function. And it actually happens to be the high mobility, low mobility mixture, the PA2, PA1 peptide amphiphile mix. And in figure K, this is actually displaying, especially in the green, remyelination of these neurons. So not only do we actually get new neuronal growth, but they're able to even remyelinate themselves. Without myelination, it's not really possible for the transduction of the electrical signals through the neurons to happen. So we really have some promise coming out of these results that function is possible with our PA1, PA2 treatment. All right, so one of the other things that was hypothesized and investigated to change after peptide amplifier treatment was actually the reworking of the vascular system at the injury site, which is also damaged along with the neural tissue. So our same now forerunner injection treatment, PA1, PA2 mixture, compared to just an uninjured mouse's vascular network, it's getting close to mimicking what normal tissue would look like. In the other treatment, just the PA2 mixture, we do see some vascular regeneration in red there, definitely not as significant as PA1, PA2. And in the sham, we hardly see any type of vascular network present. So obviously you can see the network in red, which is nice, but they just did this really the same way with all of the other in vitro methods, where they're just tagging specific proteins now for the vascular system instead of the neural system. And they find that there's both increased density, length, and branching of these vessels. So being able to also restore the vascular network at the site of injury is going to greatly increase the chance that we restore function to hind limb mobility. And that is actually what we get to look at next. So in these live mice subjects, we see some really exciting behavioral changes at that 12-week mark. So figure A is just some still shots of the mice from different treatments, the sham control, and then our PA2-1 mix, the PA only mix. So these are different possibilities of position during the walking pattern of the mice after 12 weeks. But then actually looking at C, this one's pretty exciting because they can footprint the mice and actually look at their gait patterns. So this first shot right here is just going to be like a normal gait pattern of a mouse. We see in the sham control, there's no footprints at all, and actually there's just dragging lines where the mice are pulling along the limbs that aren't moving. In our PA1, PA2 mix, the forerunner, we don't see any indications of dragging at all, which is really fascinating. There is just clear, distinct footprints, and there's a very uniform walking pattern that actually arises after the 12 weeks of treatment. And then in our other, the PA2 only treatment, there is some success. There is some footprints observed, but we still have some dragging. So there's not complete restoration of function observed with that treatment. And then the last one's just a control without the growth factor. And we see pretty much the same thing, that there's some footprinting, but a lot of dragging still there. In D and E, these just kind of plot out the actual stride length in D and then stride width in E. So in the red, this is our forerunner treatment. Number one, it is definitely the highest in terms of length and width, but it's also interesting that the cluster is a lot tighter, more in the stride length, not so much in the width. But the tighter clustering is just letting us know that this is a pretty uniform pattern. So we see a very nice restoration of function with the PA1-2 combo treatment. All right, so that is all I had to share today. I hope you guys learned a lot. I learned a lot. I felt very lucky to be learning a lot about this. I find it just fascinating with the level of success that they were able to achieve with all of these conditions in the way. And especially, there really has not been a new and exciting therapy for spinal cord injuries since the 80s, really. So this could be a turning point in this disease. The research team is currently at the moment trying to apply for FDA human trials to begin. It is going to have to be really meticulously studied. It's probably going to be a little while until we hear anything about this actually giving us a favorable result in humans. But I'm going to keep watching out for it. So thank you guys so much for being with me today and hearing me out. I want to give a quick shout out to my whole comprehensive therapy department for always supporting me through like the 3 years I've been here and really hyping me up for my presentation today. And everybody that I've run into at Insight too has been wonderful. So I'm just glad to be here. And thank you. If anybody has any questions, I think we're going to start now.
Yep. So at this point in the presentation, I'm going to give you the option to unmute yourself and ask a question if you'd like, or you can type it in the chat box and I can read it for you. But to kick things off, I did have a question myself, Bree, for you. How long after the injury would you begin this treatment?
Yeah. Yep. So the only duration that was tested in this study was just 24 hours from surgery to injection. But obviously, I think that's more difficult to accomplish in humans. So I'm not sure what range of between injury and injection is going to be tested in humans. I'm hoping that they give it a really wide range so that hopefully we can see that this works after long periods of time or short periods of time.
Got it. Awesome. And again, at this point, you guys can unmute yourself if you feel. I also will read any questions off in the chat box. Just wait a few minutes. And I'm not sure if you can see the chat box, Bree, but Christian Schultz asked, does it matter if it is partial or complete spinal cord injury?
I like that question. I wouldn't think that it would matter because these treatments are going to elicit differentiation and regeneration to the affected area. So regardless of whether it's partial or complete, I don't think it should matter too much. I do know in the study, though, if the mice were able to have any functioning of hind limb functioning after the surgery, they did not experiment on them. So there's not really data from here to support it, but I'm sure that it would work for both.
Okay. Hope that answers your question, Kristen. Is there anybody else with a question out there? All right, looks like that is it. Oh no, looks like Danielle Chadwell.
Yeah. Yep. So
go ahead.
Yeah, they haven't set a date yet for when these can begin because they're still looking actually for the FDA approval. And this study was only published, it's barely a month old, so it should be some time before there's any announcement for human trial candidates and such.
It's like you're getting a lot of praise in the chat, Brianna.
Not needed, but thank you guys, seriously.
Thank you again, great job. Everybody make sure you follow our social media platforms. And that, I believe, concludes our presentation this month. And actually for the year. So we will see everybody in January. Happy holidays. Thank you again, Bree.
Thank you guys so much. All right, everybody have a good rest of your day.